So next we'll discuss thermal dynamics in terrestrial and aquatic habitats. So these habitats have different temperature dynamics. This is because the main medium for terrestrial habitats, which is air, has very different physical properties compared to the medium in aquatic habitats, which is water. So let's first discuss the thermal properties of water. By the way, sorry for this picture. It's not really related to this topic. I put that there as a placeholder. I forgot to change it. Anyway, so water is thermally more stable compared to air. This is why water in general is less prone to temperature fluctuations compared to air. So this is due to three different properties. The first is specific heat. The second is the latent heat of vaporization. And the third is latent heat of fusion. Okay, so specific heat, this one, refers to the capacity of a substance to absorb energy without the substance changing its temperature. Okay, so water has a higher specific heat compared to air, which is why it's harder for water temperatures to fluctuate compared to air. So if you're having a hard time visualizing it, imagine that you have a heat lamp and you're shining it on letter A, one bottle, which has water, and letter B, a bottle that's empty, it only has air. So you would expect the temperatures to fluctuate less in the bottle with water. It's precisely because of water-specific heat. Okay, so it takes about 1 calorie of energy to heat up 1 cubic centimeter of water by 1 degree. So remember this ratio. 1:1:1. 1 calorie of energy to heat up 1 cubic centimeter of water by 1 degree. Next, let's discuss the latent heat of vaporization. This one. Okay? So, the latent heat of vaporization refers to the heat absorbed by a substance when it evaporates. So, for example, when water evaporates, then it absorbs heat from its surroundings, which then cools down the surroundings. So, this means that water has a good latent heat of vaporization value that it allows environments to be more stable, precisely because it absorbs heat from its environment when it evaporates. Although it's not just water naman that does this, okay? But what's special about water is that the latent heat of vaporization value is pretty high compared to other substances. So when 1 gram of water evaporates at room temperature, it absorbs more or less about 580 calories from its surroundings. Okay? So this effectively cools these surroundings, allowing the temperature to be stable even in conditions where water evaporates. So even in dry conditions or very hot conditions. Finally, we have latent heat of fusion. So latent heat of fusion refers to the heat released by a substance when it freezes. Okay? So when 1 gram of water freezes, it releases about 80 calories to its environment. What does this mean? So when water freezes, it then prevents further freezing of the surrounding area because of the calories that are released. Okay? So this is advantageous for organisms that live in these bodies of water, since obviously they can't really survive in, just ice itself. Okay? So just a short review for specific heat. Remember, 1:1:1. For latent heat of vaporization, 1 gram of water, when it evaporates, absorbs around 580 calories from its surroundings. Finally, for latent heat of fusion, okay, when 1 gram of water freezes, it releases about 80 calories. Don't forget this. So temperature also affects how well organisms perform in their respective environments. We've already mentioned this previously with the protein denaturation and enzyme activity example. So there are other ways by which temperature affects how organisms do in their environments. So let's first discuss the relationship between temperature and photosynthesis. Okay, so optimal photosynthetic rates happen at specific temperatures. Of course, extremely high or extremely low temperatures impact photosynthetic rates negatively. So I want you to look at this graph here, the third one. So it somewhat resembles the graph that I showed you regarding enzyme activity earlier on. So it peaks at a certain range, and anything above or below that range sees a dip, right, in photosynthetic activity. Sorry about this. Of course, the optimal range of temperatures for photosynthesis varies from plant to plant. There's no one-size-fits-all temperature, okay? So plants are usually suited to the temperature ranges that they're adapted to. For example, plants in a temperate forest photosynthesize better at a lower temperature compared to plants in a desert. Those in deserts, on the other hand, are more suited to high temperatures, so they photosynthesize better in hotter areas. So, let's discuss bacterial activity in relation to temperature. So you already know from microbio that bacteria are cosmopolitan in terms of distribution. But however cosmopolitan bacteria are, each species still has a certain range where it functions best. Again, the optimal temperature range depends on the species or strain of bacteria that you're looking at. Okay? I'm sure you've already heard of these terms before. So we have special classifications, special names based on the temperature range that a certain bacterial species grows best in. So, for example, we have psychrophiles. Psychrophiles thrive in low temperature areas. Mesophiles, on the other hand, usually thrive in or near body temperatures. Here, as you can see, more or less, the range of around 36, 37. And then we have thermophiles, which thrive in high temperature areas. And finally, we have hyperthermophiles that thrive in very high temperature areas, usually temperatures of above 80 degrees Celsius. So the survival of these organisms in their environments also has to do with the macromolecules that they have. For example, psychrophiles, again, the ones that thrive in low temperatures, have antifreeze proteins that lower freezing temperature, which then allows the cell to still function in very cold areas. For thermophiles and hyperthermophiles, on the other hand, the structure of their macromolecules have modifications to allow additional covalent and ionic bonds. These additional modifications make their structures more stable to allow them to, somewhat resist denaturation or having their structures compromised even at higher temperatures.